So welcome back to our webinar. Today we will talk about the recombinant protein expression, and we will talk about the key challenge that during the process and then how can we solve the problems. So for today's webinar, there will be three parts. The first part will be a short introduction to the current protein expression system that in the market. And then we will talk about some key common challenge that we have during the process, and then how can we solve them. And then last part will be some useful database for researchers to get the protein information and the structures. So we all know that proteins, they are fundamental biological macromolecules within the cells, and definitely they have important roles in the biological process during the life activities. And then basically the process can be like, you need to identify the genes that encoding the target protein, and then you know the amino acid sequence for the protein, then we can generate the CDA from the mRNAs. Later, we will insert the gene into the proper expression vectors. And I want to remind that when we choose the expression vector, we need to also consider the expression system that we use, because in my opinion, these two, they need to be suitable for each other. Then later we will screen the positive clones and then perform the expansion of the plasmid. Then later the last step will the purification of the proteins. So in CUSABIO, we have 5 different expression systems, and then they are the common expression system that we can find in the market. So for example, we have the E. coli expression system, and then for the eukaryotic system, we have the mammalian cells, the baculovirus, and the Reproducibility. Besides these 4, we also have the in vitro E. coli expression system. So very, very brief introduction to the prokaryotic expression system. So to be simple, the common used one is the E. coli expression system, because they are very easy to operate, and then also the expression efficiency could be high and also low cost. So meaning that this system is cheaper than all the others. And then because during a long ago, people already developed different plasmid for this one. So they have been already some classic plasmid in the market for researchers to choose. And then talking about the plasmid, usually they will have different core elements inside the plasmid. For example, we have the original of replication, the antibiotic resistant marker, promoter, multiple cloning site, and also the fusion text that can link to the terminal of the target protein. So you see, they are pretty classic. And then for the eukaryotic expression system, there are a lot. For example, the Petrapastorist. So this one is also kind of like a pretty popular one, because comparing to the previous prokaryotic E. coli one, this one, they can be applied to express more complex protein, which requires some post-translational modification. And then also we have the mammalian cells in this type of expression system. Mammalian cells, they make the real biological environment. So I would say that they're pretty suitable for many complex proteins. For example, the transmembrane protein. Transmembrane protein, they usually have they require a complex post-translational modification. So to ensure that the proteins that we express, they are bioactive, so they can be applied in the drug market. So, and then except the previous 2, the last one I will introduce is the cell-free expression system. It refers to the protein synthesis that carry out in the vitro. So meaning that you can use the cell lysate containing the essential component and then add this cell lysate inside to the system. And then the protein will be expressed in this fixed system. And the cell-free protein, this technology, they have been applied to the preparation of different types of the protein. The common one, the type of the protein is the toxic protein. because toxic proteins, they are usually they are harmful to the host cells. So people choose the cell-free expression system to deal with them. But I want to introduce is that in CUSABIO, we also use the cell-free expression system to express the transmembrane proteins. So as you see here, I have two examples. Both of these two proteins, they are the transmembrane proteins. So CCR4 and the SLC7A11. So, and then we already confirmed that these two proteins, they have a bioactivities. In this slide, this one is a table summarizing the advantage, limitation, and the suitable proteins that can be used with the expression system. So to be simple, E. coli, low cost, simple operation, and also the dwelling time can be short, but the misfolding is not guaranteed, especially for the complex protein. And comparing to this one, the mammalian cells is much better, but the cost can be high and also the cycle can be long. And also regarding the protein yield, with the mammalian cell expression system, the protein yield will be, not as expected as the other three. And for the cell-free expression system, I talked a lot, but I want to highlight that with the cell-free expression system, it is able to incorporate some unnatural amino acid to the proteins. So it's quite a benefit to some specific project that researchers want to do. So then let's go to the part 2, the focus today, about the key challenge and the solutions during the protein expression. Here I list some of the typical challenge that researchers have during their experiment. Low expression and the protein solubility is poor, and also the protein is not bioactive because the misfolding, and also in E. coli it's easy to see the formation of the inclusion bodies. Before I talk about some challenge that I will talk later, here I want to highlight the selection of the protein expression system. Here, as you see, this is the decision scheme for the gene expression system selection. The basic idea here is that you want to know the features of the protein. You need to know that is your protein come from the prokaryotic or eukaryotic? Because for the eukaryotic proteins, if the proteins require complex the post-translational modification, then with the E. coli expression system, you can see that there will be a lot of problems if you choose this system. Like, even though that I know in the market, they already have some commercial strains that can deal with the protein, but I will say that it's hard. Because even with the commercial strain, they enhance the ability to form the disulfide, but not all the type of the post-translational modification. So then in this case, I will recommend to just directly use the eukaryotic expression system. Then based on the projects that you have, based on like the amount of the protein yield, based on the cycle of your project, then you can choose among these 3 expression systems. If your protein, they require complex the modification, then it's highly recommended to use the mammalian cells. If your project require to have a very high protein yield, then is the expression system may be a good choice. So see, be smart when you choose the protein expression system. I think if you do this, you will avoid a lot of future problems in the project. And here I have some common causes that for the challenge, and I'm not going to introduce one by one. What I want to highlight in this slide is to say that for some type of the key challenge, actually the causes can be the same. So for example, the lack of the chaperone and the proteins tend to aggregate together. They are the common causes that for different type of the challenge. And also the culture condition and induction conditions. These are very common causes that for the inclusion body formation, and it's pretty common to cause the low protein expression. So meaning that the optimized the protein expression method will mainly focus on the strategies to optimize the vector. And actually, this is the existing optimized the protein strategies that people should choose. So in this optimization, we can optimize the promoter. So change your promoter from weaker to the stronger one. And also you can do a simple codon optimization. You can do a smart choice in the host cells. And also you can optimize the culture conditions. So all this, they are kind of like, I would say, included in the smart strategies to choose. Here is a simple scheme about the plasmid. Usually before the start codon, we have the promoter and the ribosome binding site. Then for the fusion tags, you also have a lot of choice. You can select the solubilizing tags. You can also put the signal peptides before your target proteins. And for the sequence, the target protein sequence, you can try to optimize the codons to reduce the real codons. So all this, I think, will significantly reduce the challenge that we talked. In this today's webinar, I will focus on the fusion tags and the signal peptides, and also show you the cases that we have performed in CUSABIO. So for fusion tags, usually they are the proteins that will linked to the N-terminal or C-terminal of the protein. And they are used for a convenient tools. They can use for the identification, the purification, and also to increase the solubilities. So his tag is a very popular tag, right? People use them a lot, and it's composed of the 6 histine residues. So with the his tag, you can use the his tag for the protein purification, and also if you need to perform the Western blot, the his antibodies will be a good choice. So the tags, I would say that they doesn't only have one purpose. Usually, like people deal with them like a lot. Except the common tags that in the market, some of the tags, there are a lot of other options. So for example, the summer tag. Summer tag in our company, we use the summer tag a lot. Here is one example. So we put the summer tag at the, I think it's an N-terminal of our proteins. And the summer tag increased the expression of the target protein. And as I showed you here, the length 4 is the purified target protein. So I know that it's not a comparison result, but because our company, we use this tag a lot, so we already did a lot of pre-test. And this is just one example from our target protein. So all in all, I will say the summer tag helped the expression a lot and also helped the solubility of the desired product. Another example about the fusion tags is the twin strap tag. So those who have used the his tag for the protein purification, more or less, I know that they have some experience about the protein purity. And also I know that the his tag sometimes is difficult to deal with the mammalian cell expression system. Then in this case, the twin strap tag will be a very good choice. The technical principle about the twin strap tag is the nature strong interaction between the biotin and the streptobidin. So the twin strap tag actually is 2 tag. So we have the 2 strap tag 2, and between these 2 tag, we have a short linker linked this tag together. The benefit for the twin strap tag, there were some others, like the purification process. When you use the strap tag system, is mined. So the purification condition is mined, and also the purity of the target protein is high. And it's compatible with multiple expression systems, so not only limited to 1 or 2 systems. So one example from our company is about the protein X. Before we have the his tag at the C terminal, then later we replace the his tag with the twin strap tag. And as you see here, for the protein X, the protein yield increased a lot. It's 17 times higher. And another case is about another protein, the protein Y. Before the change, we only have the his tag at the C terminal, but later we put another chain strap tag to the N terminal. Then for this, and then after the modification, this tag have this protein have two tags, and also the protein yield increased a lot. Overall is 16 times yield. So you see, the protein yield increased. And also I want to highlight the purity for both of these proteins, they are larger than 90%. So you see that it's a very easy modification, but the result is pretty good. Like the yield increased, the purity increased. Then another solution today I want to introduce is about the signal peptide. Signal peptide is similar to the fusion protein, but I know the functions are very, very different. So signal peptide usually is direct the nascent protein to the specific serial component, the environment. So for example, this is a very short illustration about the secretion system. And I want to highlight the PEL-B. So PEL-B is commonly used in the E. coli system. So with the PEL-B signal peptide, it can transport the proteins to the periplasma. So we talk about inclusion bodies can easily formed inside the E. coli. And I think it's basically because that the recombinant proteins, when they located in the cytoplasma. The cytoplasma is a very reducing environment. So with the reducing environment, the disulfide bonds cannot be properly formed. But with the PEL-B signal peptide, the nascent protein can be transported to the periplasma. And the periplasma is an oxidative environment. This environment can help the proteins to form the correct disulfide bonds. So the proteins that you get will have the correct structure. So they will not that easy to form inclusion bodies. So this is a very good example showing the importance of the signal peptide. Except this, here's another case that about the mirroring Ig kappa chain leader sequence. Let's just simple say it, the kappa signal peptide. This type of peptide is very suitable for the mammalian expression system. And I think already has been demonstrated in multiple studies saying that the expression level of the proteins will be changed if you can replace the signal peptide. And the case that I'm showing here is a very good illustration. So see, for the protein A, we know that is itself have a signal peptide. But we did a pre-experiment test comparing the self-cleaving signal peptide with the kappa signal peptide. In the pre-experimental test, the results saying that the kappa signal peptide can increase the proteins. So we continue to use the kappa signal peptide, and the final protein yield confirmed this. So overall, for protein A, if you use the kappa signal peptide, the yield is 2 times higher. However, this is not always true, because for the protein B, another case, we got a very different result. For the protein B itself also have a self-cleaving peptide, and we did the pre-experimental test, the same test, comparing the self-cleaving peptide and the kappa signal peptide. The result is very different from the protein A. With the self-cleaving peptide, the protein yield is much, much higher if we, comparing to use the kappa signal peptide. So I know it's a little bit, very interesting to see very opposite result for protein A and the protein B. But I'm not saying that which type of peptide is very good. What I want to highlight here is that to find the optimal signal peptide will help you to have a very good result. And also be sure to have a pre-experimental test, because from the pre-experimental test, the result, you can always find the optimized condition before you scale up your expression experiment. Here then will be another introduction about some classic vectors. So I mentioned that during the past research, researchers already developed a lot of classic plasmid. So it's be good to making use of this existing plasmid. Then you can just make some corresponding optimization according to your protein's characteristics. So based on the different characteristics, you can always make some change. And this work will be much easier than you start your project from making a very novel plasmid. So use the list one and add some feature. Because as I shown here, in our company, we have the plasmid, the peak 9K, and then based on this classic plasmid, we add another SummerStar tag. So after we add this tag, we just directly make a short modification, and then all the result looks much, much better. So here are some real data from our target, our project. As you see, the last line is the purified protein, the yield much, I think they are satisfied. So be good to use some plasmid and make the optimized change based on that, and the work will be much easier. The last part for the key challenge is about the codon bias. Codon bias, they refer to the differential usage frequency of the codons that encode the same amino acid. So for example, for the leucine, we know that leucine, they have the 6 different codons, but we know that in E. coli, the CUG is the most frequently used one. And because less frequency, it can affect the translation efficiency. And behind this, I think one major reason is the tRNA abundance. During the translation, the TAR abundance will affect the translation speed because for each codon, they need to wait for the tRNA and then they start the translation. So high frequency codons, because the abundant tRNA, they will be rapid ribosome binding. So the translation rate will be quick. But if you use the real codons, then the low tRNA abundance can cause the ribosome stalling, so the speed slow down. And the strategy to deal with this is quite simple, and people did a lot before. You can just directly introduce the synonymous mutation with the most abundant codons to increase the speed. And I think another benefit to do this is that you can adjust the GC content. So here is a very simple illustration. So for ARG, after optimized, the C will change to A. So you see the GC content lower down. And actually, you do not need to do by yourself. They already have a lot of online tools, and then they are free. So you can find this free online tools and then let the tools to do the correction for you. I think before I used the vector builder and IDT a lot. They will let you to choose the host expression system. Then you just put your amino acid into the box and then click the optimize. I think after several minutes, then you will get the result. After part 2, let's go to the part 3. So the database. Usually the database, I think they are very strong tools before you start the project. I totally agree that before you start a new project, you need to do a lot of spend a lot of time to prepare the project. So one preparation is to use the database to get the protein information and also to perform the protein structure prediction. This database, they are strong because they correct and organize and then store a large amount of the protein data there. So this information, common information will be the protein sequence, protein structure, protein function, and the protein-protein interactions. So for example, the BLAST, definitely everyone knows that. If you have a unknown amino acid sequence, you can just BLAST them, and then they will give you a result like which similar proteins that they are close to. And UniProt, everyone know that. UniProt is a very classic protein database. And I think in my memory, based on my past research, I think the UniProt expand a lot, especially during the last 2 or 3 years. The database is not only store the protein information, they kind of like linked to some other database. So meaning that only just with the one UniProt database, you can have access to a lot of the protein information that you can find in other database. So one example here, you'll see that in the UniProt, there are a lot of sections here. So they will have the post-translational modification information, and also they will give you the disease association, and also you can have the information about the protein structure. If you take a look at the feature viewer here, you see that they even have AlphaFold, meaning that you don't need to go to the AlphaFold database. In the UniProt, they will give you a short overview of the protein structure. So all in all, what I want to say is that I know that in the market, there are a lot of different database, but I think if you just focus on one and spend some time to dig one database, it's enough. It's kind of enough to start a project. And then you can try to expand to some other database later when the project moves on. So last here is some of the database that I used before. So for example, here is the XPRZ database. I usually use this database. So you just directly put the amino acid into the box, and then they will give you a prediction about the molecular weight. So we all know that after we purify the protein, we probably need to run the SDS phage or Western blot. Then you want to know that what size I'm expecting to see. So molecular weight prediction will be a very good information before you perform that experiment. And also here, the electric point. So this one is very good to have an idea about the pH of the buffer. So we know that during the purification process, sometimes we need to optimize the pH. And this one will give you a very good starting point to use the appropriate pH during the purification. And another database that I use a lot is the AIU PRED prediction. So for a protein, the structure state of the protein, they have the ordered domains, but they also have some intrinsically disordered protein regions. So for this one, the 0.5 is a boundary. So it serves as the boundary to determine whether this region is disordered or ordered. If the value is larger than 0.5, I will say that this region is a highly disordered domain. And if it's less than that value, this part is structured. So it's very helpful that if you're not trying to express the full length of the protein, like if you just want to express the important domain of your protein, then probably you need to delete some of the sequence. Then based on the prediction result, like for me, for this protein, I will try to delete the N-terminal part because this part is a disordered part based on the prediction. So see, it's a very good tool to kind of like know your protein structure. And the last one that I use a lot is the prediction about the signal peptide. I know I talk a lot about the signal peptide, but this database is strong. It's pretty powerful. So see, it can give you a prediction about like what the possibility that this protein have a signal peptide. For this one, the possibility is almost 100%. And did you see the dashed line here? So the dashed line is the cleavage site that the database predict. So meaning that before the dashed line, this a little bit more than 20 amino acids, they are the signal peptide for the proteins that you put. So it's very strong, strong tools. So I would say that with the open source database, it can make the protein expression to be less challenging, and then also, know more knowledge means that less problems. Last, please give me several minutes to introduce our company. So we are CUSABIO, and CUSABIO means customer satisfied biology reagent. We are specialized in the transmembrane protein expression and antibody screening. So for the here is the protein quality control platforms that for our express the protein. We perform the SDS phage to confirm the protein purity. And we also use the SPR or BLI to verify the affinity. And for some of the drug target, we use the FACS and the functional ELISA to verify the activity. For some complex proteins, we also use the mass spec to verify the sequence accuracy. So all this, they are the control platforms that we have in for our expressed protein. Here's a table about the top selling proteins during the past years, and they are all drug target proteins. They have been confirmed with the multiple platforms, and they all have a high biological activity. Except the recombinant protein, we also have other product lines. So we have the ELISA kit, we have the antibodies, we also have the molecular biology product as well as the small molecular antigens. Our company the products has been exported to more than 80 countries, including Europe, America, and Japan, Germany. We have a very good cooperation with a lot of famous companies, for example, the Pfizer, MSD, Zinker, and Moderna. And thanks for listening. If you want to stay connected with us, please scan the barcode here and follow us in the LinkedIn. We can have more communications there. Thank you so much.